Mr. Chapman here. In this video, I'm going to model the technique we use to create recombinant DNA. I'm going to start with two different pieces of DNA from two different organisms. A strand of human DNA with the gene of interest highlighted on there in green, and bacterial plasmid. This is the little ring of DNA that I'm going to insert the human gene in. So the first thing that I do is identify the gene of interest. In this case, I'm going to use as an example the gene that codes for the hormone insulin. Insulin is used in humans to control blood sugar. So I'm going to snip that gene out. The gene is located here highlighted in green, and I'm going to snip that out. In this case, I'm going to use scissors because this is paper. But if I were dealing with real DNA, I would be snipping this out with a restriction enzyme because it cuts at a certain spot. And that certain spot is a sequence of base pairs. In this case, a double C, double G base pairing. So I've snipped that gene out of the human DNA, and now I have these lone base pairs that I can splice into the bacterial plasmid. Now, if you notice, the bacterial plasmid has the same pattern, so I'm going to use the exact same restriction enzyme to cut that bacterial plasmid so that I can splice this into it. The bacterial plasmid is now opened up, and the complementary bases will bond together. If this were not paper, I would have to use another enzyme that would essentially glue or tape these back together, or cause those complementary bases to come back together. I'll do that in just a sec. And now what I have is a bacterial plasmid with the human gene for insulin inserted into this. Okay, this would be called recombinant DNA because it contains DNA from two completely different organisms. And what I then could do is insert this back into a bacterium, or just simply allow the bacterium to reproduce by asexual reproduction. It happens fast and furiously. And then all of the bacterial cells that come from that cell that had the recombinant DNA inserted in it would have this same recombinant DNA. They then would express this gene and produce the protein or the hormone insulin, which I could use to have I could employ to have little bacterial insulin factories. So the bacteria would then produce insulin for human use. Okay. And it would actually be human insulin. So then I could use that to give to people with diabetes, because people with diabetes do not have the insulin or do not produce the insulin that they need to control their blood sugar. So this would be better than like insulin from a cow or a pig because there'd be less chance of allergic reaction or other side effects. It'd be way cheaper to employ bacteria as insulin factories than it would be to extract insulin from cows or pigs or humans. And there's ethical concerns with that as well. I can't just go around taking people's insulin from their bodies and giving it to other people. That just wouldn't work out. So essentially, we've made a really effective insulin factory. And this could be done really with any protein that genes code for in all different species of organisms. I hope this helps you understand the process of forming recombinant DNA. I'll see you later.